Subscribe to our channel and hit the bell icon so that you know when live we go. Hello and welcome to RAU'S IAS DNS session. Today we are going to take one article from today's Science and Tech portion of The Hindu, and we shall discuss three explained articles that appeared in the newspaper last week. This is a news article from today's The Hindu from the Science and Tech page. Fruit fly, novel method to study nuclear metrics. The article basically is on the importance of studying nuclear metrics. And they have come up with a novel method to study nuclear metrics in a more powerful manner. See, every cell that makes up an organism contains a copy of its genome. Where is the genome? The genome basically is the sequences of gene in the DNA. And where is the DNA? DNA is in the nucleus of the cell. Inside the nucleus, there are 23 pairs of chromosomes. But the chromosomes are not floating inside the nucleus, just like the cell organelles are actually floating inside the cell. The 23 pair of chromosomes are packed in special ways with help of a structure known as nuclear matrix. The nuclear matrix gives an organization, an architecture to the nucleus. So they help the chromosomes to be aligned in a particular manner. Now, this is very important that they be aligned in a particular manner because you understand that every cell has DNA and the DNA of every cell are the same. And yet the cell functions differently because you understand that DNA is a double helix and it's very, very long. And this double helix has different parts that encodes different types of functions. For example, consider this section of DNA that is encoding the creation of protease protein. So if the protease has to be produced anywhere in the body, transcription will happen. This part of encoding will be copied and will be taken to ribosome, which is the protein manufacturer, to produce protease. But you understand that the production of protease is not required everywhere. It will be mostly required in stomach for digestion of protein. So this section that is encoding the production of protease should be expressed in stomach. It should not be expressed elsewhere. For example, in brain and other parts of the body, they should be hidden so that an accidental transcription does not happen. Otherwise, it will dissolve that part of the body. So, depending upon the function of the cell, those part of gene should be easily accessible for transcription. And if that part of the gene is nothing to do with the function of the cell, they should be tightly and very safely hidden. So although every cell has 23 pair of chromosomes, but the arrangement of the chromosomes are different in every single cell. And that arrangement is decided by nuclear metrics. Nuclear metrics in biomolecular science are famously called as scaffoldings. Scaffoldings are kind of ladders, but it's three-dimensional, so you can actually go up the height from any direction. This is a very basic rudimentary structure of a scaffolding. This structure, this scaffolding, this structure of nuclear metrics, it's very important to study them. That will enhance our understanding of function of cells. And in the embryo, when there is single cell, and from there different cells develop, in that cell division, different cells for different part of the body are developing. And now we understand that the nuclear metrics of every cells are different. So studying nuclear metrics in the embryo helps us understand the developmental process in the organism. The traditional way of studying it is to bring the nucleus out of the cell and then treat it with an enzyme that digests all the DNA. And then they wash it with a solution of high salt concentration so that the DNA-protein and protein-protein interactions are broken. So basically the DNA gets out of the scaffolding, the basic structure that holds it up. After washing away, what's left is the scaffolding, the nuclear metrics, and then they used to study nuclear metrics. But now they have devised a way to study different cells without taking the nucleus out of it in the embryo together. There are 220 different types of cells in the body. And you understand that they all contain identical genomes. They are the same in the neurons, which are used for thinking and transmission of signal. The same genome is present in the liver. The same genome is present in the intestine. The same genome is present in the brain. But yet different part of the body functions differently. So the packaging of the genome, the DNA structure in the chromosomes must be different. And to hide particular genome which should not really be active. For example, we just discussed that if protease would be allowed to be active in brain, they will digest all the brain cells. because there, like the intestine, we do not have the protective epithelium tissue. So the genome, despite carrying all the genetic material, is packed such that some genetic material is hidden in such a way that it is never seen by the transcription machinery. And this can happen only if packing is done in such a way that something becomes so dense and interior that it is not seen. Most human cells spend 90% of their time in a state called interphase. Interphase is the phase of the cell cycle in which a typical cell spends most of its life, and this is the daily living or the metabolic phase of the cell, in which the daily life of the cell goes on, which is to collect nutrients and metabolize them, grow itself, and then replicate the DNA in preparation for mitosis, the cell division. So the cell basically prepares protein and carries out the intended function prior to mitosis. So ordinarily 90% of the time of the cell is in interphase. In each of these cells, there is a nucleus around 60 micrometer in diameter, and that is around 1/10 of the width of a human hair. Within the small nucleus, there resides a nucleolus. Nucleolus is the largest structure in the nucleus of any eukaryotic cell. And apart from nucleolus, there are proteins and around 2 meters of DNA, splitted into 23 pairs, totaling 46 chromosomes. Each chromosome comprises of 2 territories. One of the territory, let's say territory A, sits closer to the nucleus and it is less densely packed, hence allowing easy access to the gene so that they can be expressed easily by the transcription machinery. The other territory, let's say territory B, that sits towards the periphery of the nucleus and is more densely packed, and hence it is mostly inactive. Within these inactive territories, similar kind of genes gather together, so that similar kind of functions can be performed simultaneously. Technically, this is called as topologically associated domains. If we look closer into one of these microdomains, there would be many smaller nucleosomes. Nucleosomes basically are DNA wrapped around another structure. So a small segment, a basic fundamental unit of DNA that gets wrapped around another structural protein, that is called as nucleosome. A nucleosome is made up of 8 histone proteins. And DNA wraps around these 8 histone proteins roughly twice. And this wrapping around of DNA around these histone proteins basically gives the structure to the chromosome. So these proteins around which the DNA are getting wrapped are kind of forming a scaffold. These proteins give structural framework to the DNA, and this is called as nuclear matrix. Now, of course, there has to be some kind of interaction between some segment of DNA and some amino acid part of the protein in the nuclear matrix. The region where the interaction between the DNA and the structural protein happens is abbreviated as SMARS. Basically, it means scaffold or matrix, attachment region. So S/MARS, matrix attachment regions. We will not get into the further technicality of this as to how the attachment between the DNA and the scaffold protein happens, but you would understand that there would be some DNA binding proteins in the structural protein, the scaffold, the nuclear matrix. And that one particular protein is informally called as AT hook. And you understand that there would be nitrogen-based cyclic molecules. There would be some amino groups. And a hydrogen bonding must be forming between different groups, just like it is happening between the 2 strands of DNA that helps produce the double helix model. Some of these interactions between the DNA and the nuclear matrix are very stable, very, very long lasting. This is how the chromosome structure gets stabilized. But there are also less stable interactions. Because you understand, when the transcription has to happen, the bonds between the 2 strands has to be broken in the DNA. And also the interaction with the nuclear matrix has to be broken so that the transcription protein comes and can do the reading from the DNA. So there are some inhibitors which are activated when the transcription is supposed to happen and the bonding between the DNA and the nuclear matrix are broken. So after the transcription has begun, the SMAR elements dissociates. so that the gene can pass through the transcription machinery. So this combination of consecutive and transient SMAR interactions with the nuclear matrix allow the compaction of 2 meters of DNA into a tiny nucleus while gathering functional, and all this with gathering of functionally relevant genes near one another and also regulating the transcription of these genes. These things are possible because of the structural design of the nuclear matrix. Now let's do this explained article from this week. The One Nation One Fertiliser scheme, the government's logic and some immediate risk. The Ministry of Chemicals and Fertilizer has recently announced a new scheme, Pradhan Mantri Bharatiya Janurvarak Pariyojana. Under this scheme, it has been decided to implement One Nation One Fertiliser by introducing a single brand for fertilizers and the single logo for all fertilizers. See, the way it will work out, all companies will be allowed to display their name, their brand, their logo, and all other relevant information only on 1/3 of the space on their bags. On the remaining 2/3, the Bharat brand and the Pradhan Mantri Bharatiya Janurvarak Pariyojana's logo must be shown. So there will be a single brand name for urea, for diammonium phosphate, for muriate of phosphate, for NPK based fertilizers, and they all will have Bharat as prefix. So effectively it should be called as Bharat Urea, Bharat DAP, Bharat MOP, and Bharat NPK fertilizer. See, the rationale given behind this move is that the government incurs huge expenditure on fertilizer subsidy. However, the fertilizer companies continue to sell the fertilizers through their own brand names without giving any due credit to the government. And hence, the new scheme will enable the government to take the due credit. But there are criticism and concerns against this move because now the branding can be done only in 1/3 of the space. So the brand name, the logo, and all relevant information, they all has to be given in 1/3 of the package space. This will reduce the scope for the brand identity. It may disincentivize the fertilizer companies to ensure good quality fertilizers, because maybe all kind of fertilizers would be considered as same. And hence this move can effectively discourage the private sector investment in the fertilizer sector. Well, this is an administrative move and there is not much to study here. But since this thing is in news, we must get back to the basics. Government of India has been giving subsidies for fertilizer to the farmers since 1970s. And the government has devised separate mechanism for urea subsidy and for non-urea, mostly the potassium and phosphorus fertilizers. For urea, the method for giving subsidy is called as cost plus method. Here the MRP of urea is statutorily fixed by the government, and the difference between MRP and the cost of production is provided to the manufacturing company as subsidy. And hence, different amounts are given to different manufacturers based on their cost of production. This method is called as cost plus subsidy. The cost plus subsidy is for urea. For non-urea fertilizers, mostly including phosphorus and potassium, the method of giving subsidy is called as nutrient-based subsidy. Here, unlike the case of urea, the market prices are deregulated. Manufacturers are given free hand to decide the market price, and they receive a fixed subsidy from the government based upon the amount of nutrient present in the given amount of fertilizer. Based upon the proportion of the nutrients in the fertilizer, per kg subsidy is calculated. See, the fertilizer subsidy burden on government is very high because government not only gives direct fertilizer subsidy, it also gives the freight subsidy. For urea, government provides almost 80 to 90% of cost of production as subsidy. Similarly, the government continues to provide subsidy to companies under nutrient-based scheme, which has been very controversial, and there were suggestions before to stop this subsidy. So nutrient-based subsidy to non-urea fertilizers, mostly including phosphorus and potassium. This also has been continued. So the fertilizer subsidy bill of the government is very huge. It is expected to be very close to rupees 2 lakh crore. This is only second to food subsidy in terms of expenditure. But direct fertilizer subsidy is not all. Government do subsidize and decide the price of the price of the fertilizers, but it also subsidizes the transport of the fertilizers. The Department of Fertilizers draw a monthly supply plan based upon consensus with the manufacturers and the importers and the distributors. This supply plan is to ensure that fertilizers are available as per requirement even in the remote areas for the sake of food security. So the supply management of fertilizers that is also subsidized. Let's look at this previous year question from the prelims paper of 2020. With reference to chemical fertilizers in India, consider the following statements. At present, the retail price of chemical fertilizers is market driven and not administered by the government. We have just seen that the fertilizer subsidy burden is very high because government decides the market price, and the difference between MRP and the cost of production is given as subsidy for the urea fertilizers. And there are other subsidies that you know very well of. So it is not market driven. It is completely administered by the government. Statement is incorrect. Ammonia, which is an input of urea, is produced from natural gas. It is correct. Ammonia has been synthesized for very long from natural gas. In this process, the natural gas, which it is reduced to carbon and hydrogen. The hydrogen is then purified and reacted with nitrogen. This produces ammonia. This synthetic ammonia is used as fertilizer either directly as ammonia or indirectly after synthesis as urea, as ammonium nitrate, as ammonium phosphate. So the statement is correct. Sulfur, which is a raw material for phosphoric acid fertilizer, is a byproduct of oil refineries. You know that sulfur is a major byproduct of oil refineries and gas processing. In fact, you know that the quality of petroleum product, it is judged by the percentage of sulfur. Lesser the sulfur, better the petroleum product. Most crude oil grades contain some sulfur, and most of them are removed after the refining process. Sulfur actually can also be mined from underground. There are naturally occurring deposits, but this is very costly. Sourcing it from oil and gas is relatively cheaper, and anyways it is produced as a byproduct. From sulfur we produce sulfuric acid, and sulfuric acid is used to produce both monoammonium phosphate and diammonium phosphate. So the statement is correct. The answer is option B. This article in the explained section has been published very recently. What is Arth Ganga? The government's new model for the river's sustainable development. Recently, the Director General of National Mission for Clean Ganga, he was speaking on the occasion of the World Water Week 2022. In his virtual keynote address, he spoke about the concept of Arth Ganga model. Before we begin any further, let's talk a little bit about National Mission for Clean Ganga. See, National Mission for Clean Ganga was registered as a society back in 2011. This was created as the key implementing arm of National Ganga River Basin Authority. National Ganga River Basin Authority was constituted under the provision of Environment Protection Act 1986. But National Ganga River Basin Authority was dissolved in 2016 because of constitution of a new body called as National Council for Rejuvenation, Protection and Management of River Ganga. This is more popularly referred to as National Ganga Council. For the management of Ganga riverbed, organization of 5 layers have been set up. At the top, there is National Ganga Council. You have to remember this is chaired by the Prime Minister of India. At the second level, there is Empowered Task Force on River Ganga under the chairmanship of the Minister of Jal Shakti. At the third level, there is National Mission for Clean Ganga. Now, this is largely the implementing body. Then we have State Ganga Council and District Ganga Council in every district through which the River Ganga and its tributaries passes. Now let's come back. Director General of National Mission for Clean Ganga, the third level body, spoke about the Arth Ganga model during the virtual summit for World Water Week 2022. Since 1991, the Stockholm International Water Institute has been organizing the World Water Week every year to address the global water concerns. But this was not the first time that the Arth Ganga model was spoken about. In fact, the Prime Minister of India introduced this concept during the first National Ganga Council meeting in 2019. Remember, Prime Minister is the chairman of National Ganga Council. In this meeting, the Prime Minister urged for a shift from Namami Gange to a model of Arth Ganga. You must be very familiar with the term Earth Shastri. You know Earth is related with economics. So, at its core, the Arth Ganga model seeks to use economics to bridge people with the river. So, although it is based on economics, but later the boundaries of the concept was enlarged to imply sustainable development with a focus on economic activities related to River Ganga. So, as part of this process, the farmers would be encouraged to engage in sustainable agricultural practices. Like, for example, zero budget farming, planting of fruit trees, building plant nurseries on the bank of River Ganga. There would be other measures that will be focused upon, for example, organizing women self help groups. Also, having an ex servicemen organizations for the implementation of programs related with Earth Ganga model. There are also plans for creation of infrastructure like infrastructure related to water sports, development of campsites, cycling, walking tracks. These would help to tap the hybrid tourism potential of the river basin area for the purposes of religious as well as adventure tourism. And the income generated from encouraging ecotourism and Ganga wildlife conservation and cruise tourism, etc., would help to generate sustainable income streams for cleaning Ganga. All these measures are classified into six verticals as is stated by the government. The six verticals under the Earth Ganga model includes these. The first, zero budget natural farming, which involves chemical free farming on 10 kilometer on either side of the river and promoting cow dung and fertilizers through the Gobar Dhan scheme. The monetization and reuse of sludge and wastewater is the second, which seeks to reuse treated water for irrigation, industries, and revenue generations for local urban bodies. Earth Ganga will also involve livelihood generation opportunities by creating hearts where people can sell local products, medicinal plants, and Ayurveda products. The fourth is to increase public participation by increasing synergies between the stakeholders involved with the river. For example, creating ex servicemen organizations. The model also wants to promote the cultural heritage and tourism of Ganga and its surroundings through boat tourism, adventure sports, and by conducting yoga activities. Lastly, the model seeks to promote institution building by empowering local administration for improved water governance. You must have some update regarding some recent initiatives taken under the Earth Ganga model. Jal Shakti Ministry has unveiled some new initiatives in the recent times. For example, Jalaj Initiative. Jalaj Initiative is being implemented in association with Wildlife Institute of India. A trained cadre of Ganga Praharis they have been created from among the local people by Wildlife Institute of India for biodiversity conservation and Ganga rejuvenation. Jalaj basically is an innovative mobile livelihood center, which basically aims at aligning the skill enhancement activities with Ganga conservation program. Recently, a MOU also has been signed between the National Mission for Clean Ganga and Sahakar Bharti. Sahakar Bharti is a non government organization which involves cooperatives. The purpose of the MOU to realize a sustainable and viable economic development. So basically, the local cooperatives will be strengthened to align their economic activities with the ideas and goals of Earth Ganga model. I am Avatar. It's a tourism related portal to promote livelihood opportunities along the river Ganga basin. The idea is to promote tourism, marketing of local products, both agricultural and handicraft products. The Jal Shakti Ministry recently has inaugurated an online portal called as CLAP, Continuous Learning and Activity Portal. It has a course called as River Champ. This online platform is for raising awareness, discussing and debating the actions and strategies for river conservation in India. There is an explained article, Jupiter like never seen before through the Webb telescope. They are referring to the James Webb telescope, and The image that you see here of Jupiter, generally you see it as a reddish giant ball of gas. But here a different color has been used to signify the different altitudes at various locations in the Jupiter. The point here is to understand why it is like never before. So we'll take an opportunity to understand the basics of telescopic astronomy and what's so special in the James Webb telescope that now we have images of the planets that we never saw before. See, first of all, you would know that the electromagnetic radiation can be divided into various bandwidth. The one having the highest energy are in the range of gamma rays, followed by X-rays and ultraviolet. And you would understand things that are hot. They would emit radiation with larger energy. They would emit radiation in this range mostly of ultraviolet, X-ray and gamma ray. So the young stars, they emit radiation mostly in this region. So the electromagnetic wavelength that will be used to study young stars by any telescope would be in this range of ultraviolet and X-rays, mostly these two. Then as you decrease the frequency and hence the energy of electromagnetic radiation, it starts to come in the visible range, followed by infrared, microwave and radio wave. What you might already know and is damn important in telescopic astronomical study is that Earth radiates in the region of far infrared. Infrared can also be subdivided into far infrared, mid-infrared and near infrared. Near with respect to the visible range. The one which is nearer to the visible light bandwidth, that is called as near infrared, then mid-infrared, followed by far infrared. Now this is the region in which our Earth emits radiation. Radiation coming from the sun brings all sort of radiation. But after absorption of radiation, the Earth emits in this region. Now the thing is, if you have a telescope on ground, if you have a ground-based telescope, then this telescope cannot use the electromagnetic radiation in this region, first of all. Because electromagnetic radiation in this region does not reach Earth. If it had been, we all would have been dead. The atmosphere actually filters out ultraviolet radiation. And higher the energy of the radiation, the faster it gets attenuated. So X-rays and gamma rays do come out of stars, but it does not reach the Earth. The ultraviolet manages to reach into the atmosphere of the Earth, but it gets absorbed. So a ground-based telescope will not have access to the ultraviolet, X-ray and gamma ray radiation coming from distant stars. The ground-based radiation can also not use far infrared because it is being emitted by Earth. So it will act as noise interference. In fact, ground-based radiation does not use infrared at all. They don't use microwave also because of cosmic microwave background radiation. Ground-based stations mostly use radio waves in association with visible light. Now suppose a telescope has to read a star. Then to read the star, you have to study in the wavelength which the star emits most prominently, which is ultraviolet, X-rays, gamma rays. So you have to go into ultraviolet region. So you cannot do it from the Earth. Or to say you cannot do it in ultraviolet, X-rays and gamma rays. You can do it indirectly through radio wave. But if you want to study young stars radiating high amount of energy, then you have to go for ultraviolet and X-rays, which you cannot do from the surface of the Earth, which you cannot do even from atmosphere, which you cannot do from elevated balloons. You have to get out of the atmosphere. You can do it from low Earth orbit. The low Earth orbit above the atmosphere will get the ultraviolet rays, to a certain extent maybe X-rays. But from low Earth orbit you cannot do in the far infrared region because the radiation emitted from the Earth will get out of atmosphere and reach in this region as well. But from low Earth orbit you can use the near infrared region. But if you go very deep into the space, maybe the Lagrange point L2, there you do not have a problem of using infrared light. So from L2 you can use infrared. This is how generally the satellites work according to their purpose. Their location and the wavelength in which they will operate is decided. For instance, AstroSat is the first astronomical mission of India and it works in multispectral bands. For instance, it works in the range of X-ray, optical bandwidth, the visible bandwidth, and ultraviolet spectral bands as well. So you would understand where AstroSat is. AstroSat cannot be a ground-based station because it cannot work in the ultraviolet region from ground. Neither it can work in X-ray region. Where is it? It is in the low Earth orbit, around 650 km from the surface of the Earth. You might be aware of Aditya L1 mission of India. It will be sent to Lagrange point L1 here. Lagrange point L1 is around 1.5 million km from the surface of the Earth. Aditya L1 is a dedicated mission to study the Sun. This is the first solar mission of India. It is supposed to be launched early next year. This Aditya L1 mission will have on board a telescope, Solar Ultraviolet Imaging Telescope. As I have told you before, to study the Sun and the stars, which radiate high amount of energy, you have to go in this region. So naturally, to study the Sun, ultraviolet region will be used. You must have heard of the famous Hubble telescope. Hubble telescope works in the region of near ultraviolet, visible, and near infrared. I told you, Earth emits in the region of far infrared. So if you use far infrared, you cannot be around Earth. You cannot be even in low Earth orbit. But if you use near infrared, you can be in low Earth orbit. And visible light you can use from anywhere. And if you're out of the Earth's atmosphere, you can use ultraviolet as well. So based on this knowledge, you can intelligently guess the position of Hubble telescope, which presently is in low Earth orbit. But very soon, Hubble telescope will be replaced by a very, very powerful telescope, the James Webb Space Telescope. James Webb Space Telescope will be working in mid-infrared region. You have far infrared, mid-infrared, and near infrared. Near infrared is near to visible light. Working in near infrared is easy because it is away from far infrared, the range in which Earth emits radiation. But if you work in the mid-infrared region, you come close to this region in which the Earth emits radiation. So if you are going to work in the mid-infrared region, you cannot be in the low Earth orbit. So James Webb Telescope is not going to be sent in low Earth orbit. It's going to be sent in L2, Lagrange point 2. This is the L2 point and this is in the shadow region. A shadow is created because of presence of Earth in between this point and the Sun. And the temperature goes very, very low, around 50 Kelvin. And this low temperature is suitable to study in the wavelength of low energy. And infrared, you know, as low energy. The advantage that James Webb Space Telescope will have by working in the range of mid-infrared is that it can see distant galaxies and stars, the one that cannot be seen by Hubble telescope. Because due to the red shift that happens to radiation coming from distant stars and telescope, the red shifting if is large, then the radiation will come in the mid-infrared region. If more red shifting happens, your radiation will keep shifting from near infrared to mid-infrared. And red shifting is more if the galaxy or the star is more distant. So for very, very distant stars and galaxies, the radiation will come in the mid-infrared region, which Hubble telescope cannot see. So James Webb telescope will see beyond what Hubble telescope can see in the universe. And for that purpose, it has to work in the region of lower frequency than what Hubble telescope was working in. Hubble telescope used near infrared region. James Webb telescope will use mid-infrared region.